Um, I hope everyone here, and it is a great moment that we can learn from these practical skills in our laboratory at Christian University. I must say that for scientists like you, studying, you need to have hands-on. You have to perform some practicals and procedures that will help you to give an answer to a given question. Now, looking at environmental microbiology as a course unit, and I would like to introduce you to environmental microbiology lab. The basic information that you need to know when you are in the microbiology lab. So it's very important before entering into the lab for us as students and staff to understand the safety precautions, safety measures. As we know very well, these microorganisms, they live in our surroundings and they are with us. They're within our body surfaces, inside our body. So some of them are pathogenic in terms that some of them can cause disease, some of them uh, cannot cause disease. So it's very important for us to know the safety precautions that we need to take in the lab as you're doing your experiment, as you're doing your test, determine or to isolate which microorganisms are in the environment. So in that case, the environment, you may want to test the quality of water. How is this water? Is it safe for human consumption or for use? Basing on the microorganisms that you know that they are actually pathogens to human beings. So this information that we get from that is that it will help us to protect ourselves as we go, and as well as also protect others who may come and use it when we. So it is very important for you to take note of this precaution. One, when you come into the microbiology lab, you must have closed shoes. You must have put on closed shoes. You do not need to come in sandals because anything can happen. You may be preparing maybe culture media or maybe carrying some equipment. If it drops down by mistake, you can get a cut, prick, so that will introduce a microorganism. So because these microorganisms, you don't see them with our naked eyes, you can't tell on the surface whether the microorganisms are there. In the laboratory being a place where some of these microorganisms can be stored, we need to take extra care. And that's why we need to put on closed shoes, we need to put on the laboratory coats that we have here. You have to put on gloves before doing carrying out any procedure. You need to put on gloves like I'm doing here. You have to put on gloves all the time when you're going to handle work. And for ladies who are having long hair, you are reminded and requested that you tie your hair back so that when you are doing procedure isolation of this microorganism, or you are doing your procedure, your test, the microbes that are on your hair will not be able to fall on the surface of the media that you use for testing. Otherwise, you will give wrong results and you will disqualify someone's information or sample that it has is not safe when actually the microbe you isolated could gotten into the surface of the medium from your hair. So it's very important for you to then Another thing is that before handling any procedure, you must follow the standard operating procedure. Why? Because there are some equipment that have to open them now, or maybe solutions or what liquid that you use in the lab, they must be having some effect that you need to take note of. So if you understand the SOPs, it will help you to avoid that kind of injury hazard. Then another thing that you need to take is the proper waste management. When you're in microbiology lab, you need to all the waste, you have to segregate them according to how they are. If it's biological, you put in the biological waste bin or highly infectious waste bin. For those which are non-infectious papers, you put in that right fold. So that means all those bins have to be. Then all the surfaces of the lab must be cleaned before and after use. You have to clean the laboratory surfaces before use, so that before use, if in case somebody had dropped this maybe sample on the bench and you were not there seeing the person, so it will help to eliminate that microbe so that you are safe. Or else, as you're leaving, you need to clean it because another person will come and leave it. Very important. Now, when you're dealing with acids and bases, you need to put on eye goggles, and also where necessary, you need to put on masks. So it is very important for you to take note of this. And if you have any allergic reaction, you have to report to the laboratory technician before, because there are chemicals that you may handle and you might be allergic to. So when you inform the person, the lab technician will guide you on how you are going to do practical microbiology. And so many other precautions that you read about. I'm not going to mention them because I think I need to take you to the, the procedure that I have here. You will read those on your own. Now, it is very important for you to know the major equipment that we use in microbiology. These equipment are ranging from those ones which are very, very heavy that are not portable, like incubators, fridges, autoclaves. So I am unable to bring them here for me. But when you reach in any lab near you where you do your practice, you ask any technician there, they will show you what an autoclave is and what an incubator is. All those equipment have got their uses that you need in your procedure. Now, for every equipment, there is standard operating procedures or the manufacturer's instructions. They give you that manual. They give you the manual that you have to read through before use. So it is very important for you to read this manual inside, all for all the procedure, all the precautions they are going to give you. So it is very important for you to read manufacturers' and standard operating procedures. Now, for in microbiology, we have culture media. Example, this is McConkey agar. This is nutrient agar. This is clad, and so many others which I have not mentioned. But now, for isolation of microorganisms, this culture media, you need to prepare them following the manufacturer's instruction, which is always specified on every media here. So you can say the language that you understand most. You find it's in German, you might find in English. So for us here, we're using English. So for this as cleared, you can see the procedure here. Down here, be able to see it. So you read that carefully and you see what is make you understand how much you should weigh and dissolve in which volume is still water. Distilled water, this is a distilled water, and normally we prepare this culture media in bottles like this. This is Scott bottle, and it has a plastic cup, but it's heat resistant, and you can be able to sterilize this. So meaning that even the water that you're going to use must be sterile. And also must be free from other ions, because these different ions affect the growth of microorganisms. You will need to weigh this, our weighing balance, analytical balance, and then you dissolve in a specified volume of water. You dissolve it in here, then you will take and then put it. So after the preparation, you will be able to quality control your media to ensure that there is no contamination. Why should you take note of this? Because if the culture media itself is very contaminated, that means even the specimen that you're going to now uh, culture on this culture media, it will give you wrong results. The fact that already this culture media are both microorganisms. It will mean the, the organism that you have isolated is not necessarily from the specimen, but it is from the culture media, which has resulted as from your contamination, wrongly handled. So that is that for how these are some of the major equipment we use. Take it aside. We also have slides which are not here. I'll be able to demonstrate to you next time when in my video when I'm doing microscopy. The slide will help you to examine the structures of different microorganisms. You will realize that you learn about gram-positive organisms and gram-negative organisms. So on the slide, it will give you that information. And why do they say this gram-positive? Gram-negative is based on the thickness of the cell wall of the different microorganisms. For the gram-positive, they have thicker layer of peptidoglycan, and then that of gram-negative, they have thin layer of peptidoglycan in the cell membrane. So that's how you can differentiate between gram-positive and gram-negative. As engineering and environmental students, you will be uh, dealing mostly with use indicator organisms that you get in your environment to indicate that maybe there is fecal contamination or not. And you're going to be using total coliforms and E.Coli. So those are the major ones that I'm going to illustrate to you how you carry out that investigation in order for you to isolate E.Coli and protocol. However, there are so many other culture media that you can use that can help you to isolate the different microorganisms. So in my next video, in the next few seconds, I'm going to show you how these are now is also a culture medium, but it's a liquid media. Is a broth, and we use this in isolation of Escherichia coli and Coliform. We rate them in ounces. So we shall be using this, but if you look at this, the color here is abnormal. The color would have been like this, less actually red. But now once it's stable like this, you realize that there is some culture media. You must have learned from the class. Culture media contains all the growth requirements that microorganisms need for their multiplication or reproduction. So it contains the carbon, it contains hydrogen, contains the oxygen, um, phosphorus and sulfur and others at a certain pH, balanced pH. So that is culture medium. And then also we have, say, here we have ethanol, absolute ethanol that we will be using this in when you're sterilizing some of the equipment. You put in a drop of this and then you burn it until you clean the surface, sterilize the surface, so that you will be able to use that equipment without any contamination. Now, on this, we have also this. These are concepts. And then also I have another one here. This is also another concept that we use. And then here I have petri dishes. Petri dishes is like it's an equipment or small equipment that we use for placing our medium for preparing our culture media, so that when we inoculate, we apply our the surface of the media on the media, then we shall be able to isolate the microorganism. Okay? So these petri dishes here are made specifically for use in membrane filtration technique. This is sterilized water you can use for preparation of culture media, both all the liquid media. Here. So I'm just going to show you how you are going to do membrane filtration. I'm going to know this is the membrane filtration unit. As this starts, realize that inside here, there is that lower area where you see that ring, shows the volume, and then on top here is another volume. So the first ring in the middle that you see there is for 50 milliliters of water, and then all that sample we're testing. Then this other last ring here, that one there, right here, is 100 milliliters of water sample. So this is a membrane filtration unit, and this is the suction pump, which is the hand pump that will help you to reduce pressure that will suck the sample fluid. We also have other consumables. Have these consumables that are used, and this is sterile pads for absorbing the liquid medium after preparation. Actually, it absorbs the food's microorganism, and then this is sterile membrane filters. They have specific size and diameter. You can see it here. This one is the pore size is 0.45 micrometers and the diameter is 47 millimeters. Okay, so take note that these things are not expired and make sure that they are not open anyway. Make sure that they are sealed, okay, and to single use on the one. So you need to have your specimen ready when you're going to, you have to prepare everything ready. Your specimen has to be there. You have to have a faucet. You need to have a sterilizer. So you can use alcohol for sterilizing your faucets. And now you are going to open up this down. And then also this one is sterilized. Sterilized. What you're going to do is open up this membrane filters. Open this membrane filters carefully. This is relaxation. Without touching on the surface. This sterile faucet, pick this. Then with a grid, this has a grid like a mark. Okay, I don't know if you're able to see the grid. Yes, you can see rows and columns. That should be facing up. So you place it on this surface. There. Okay. Then you bring your cup and remember to be doing this on the bench surface. Okay, where you don't risk fall, and you connect this pump here. Okay. Then you are going to now use sample. Let's assume that this is our specimen that we've got from the field. You are going to open this specimen and then you pour. You will pour it up to the mark that you see. If you put it at the first ring there, that means you've used 50 milliliters of the sample. And if you fill up to here, that's 100 mils. Then you start press, and it will be sucking water through until all the water is sucked. And then if there is any possibility of any microorganism in this specimen, it will be trapped on the surface of the sterile membrane filter. Here, after that, you're supposed to have pick one sterile pad and place have dispense. This is not sterile, uh, but you need to prepare a fresh one. This is an old stock. Then you will get 3 mils. You can use this pipette, but this one is that's a very small volume. You can dispense the media using a syringe, and you can open it up. All is sterile. You can get the volume of the media, about 3 mils, and then you dispense on the surface. Okay. This sterile pad will absorb the media, and then now make sure that you cap everything that you have used immediately. Place it in safe place. Okay. Now you will now open up this what you have filtered using the sterile forceps. You first sterilize it and then pick all one side like this. Then you place on the surface without touching it with your hand. Okay, and then immediately cover this. And this at this stage, you're supposed to label. Label this with the name of the specimen or the specimen number, and the same number should be on this surface. Okay. Then now after this, you take this into the incubator. I mentioned earlier that we have equipment called an incubator. So this incubator provides the right food temperature when you're culturing. So for E. coli, you need to set the incubator at a temperature of 44 degrees Celsius. Meanwhile, one of total coliform, other coliforms, you set at 37 degrees Celsius. Then you incubate this for about 18 to 24 hours. Then thereafter, you count the colonies according to the appearance. And the procedure of how what kind of colonies to count and that will show you which colonies to count. Okay, this is just showing you how you assemble the equipment and how you do membrane filtration, and then you see showing you how you can place the filtered sample on the petri dish, and then these are the colonies I'm talking about. You see there, this yellow dotted thing, these are the colonies, and then these are the ones you count, and then there are also the red ones. Okay, and according to this media that I'm saying that any yellow colonies that you see there is E. coli. Okay, Escherichia coli, and then the others are considered to be the red ones are considered to be Epicolic. So whatever you count from here is in that volume that you put. So if you put less than 100 ml, you have to calculate and indicate that you have to report in a standard way that the number of the colonial forming unit per 100 milliliters of the specimen. So you need to learn how to do that calculation. So that will, after recording the final things, now you have to clean up this equipment through a series of procedure. Do sterilization, and then again, you can now wash them, and then dry them up, and then make the next use. Finding it clean and start from. So that is that for using membrane filtration unit. And the next time I'll be now showing you how you're going to use the agar media for inoculation of specimen on the surfaces, and then you'll be able to follow. And I will also illustrate to you how you can do the weighing using our analytical balances, and then also I will show you some of the equipment that are not with me here. So thank you very much. I wish you all the best.